Welcome everyone, I'm James Nagai, currently I'm a PhD student at the Institute for Computational Genomics from Aachen Hospital University, and today it's my pleasure to introduce you our work dissecting differential cell-cell communication with CrossTalkeR. One specific cell-cell communication mechanisms occur through ligands and receptors, and motivated by the single-cell RNA-seq high-resolution, several methods attempts to infer cellular crosstalk using those interactions. Usually, those methods receives as input the ligand receptor database and a single-cell RNA-seq matrix, and by looking at the gene expression profile of each ligand and its cognate receptor, the methods are able to return a list of the statistically significant interactions. Although several methods are out, considering different aspects, most of the methods do not allow us to compare different phenotypes, and also network representation remains underexplored. To fill this gap, we introduce here CrossTalkeR. CrossTalkeR is an R package for characterization and prioritization of single-cell differential cell-cell communication analysis using network representation. CrossTalkeR receives as input the ligand receptor analysis done using cellphone DB and builds two different networks. CrossTalkeR employs node topological measures such as influencer given by out degree, listener given by in degree, mediator given by between the centrality to prioritize and ranking those nodes, which could be a cell or gene ligand or gene receptor in its phenotype. To illustrate how CrossTalkeR can be used, we revisited a collaborative work of myelofibrosis. The initial work was presented by Lane Huller et al. In 2021, and here we get the human sample of the study to show how crosstalk works. The input data is composed by 5 different cell populations, although the number of samples is low because this is from human and it's really difficult to get these human samples. We could see some changes that's statistically significant as shown by the markers on the left. In the first step, CrossTalkeR builds the cell-cell communication network. In this cell-cell communication network, the nodes represent the cell populations, the edge represents the interactions between those cell populations, and the node size is showing the relative node importance given by log's odds PageRank. The edge colors represent the expression, and the edge thickness represents the number of interactions. By using the relative node importance given by log odds PageRank, we could highlight the major players of myelofibrosis, which is megakaryocytes and neurocells, corroborating with our previous study. We have also another resolution, which is called gene cell network, but in the gene cell network, the network is complex and very difficult to understand. To overcome this, we use the node topological measures to embed all the gene cells using PCA. In this way, we can highlight the major players according to each node role, being as influencer, mediator, PageRank, or listener. And as a result, we could highlight important players to myelofibrosis, such as TGF-beta in megakaryocytes or collagen 1 in MSCs, showing increasing matrix secretion by MSCs, and also inflammation from starting with TGF beta. We also show that CXL12 is, on the other hand, of those interactions, which then shows the loss of hematopoietic support, which is really related to the control stage. More than that, we allow that the user uh, explores each of these players individually. In this way, we have as example TGF beta 1. In TGF beta 1, we see that most of the interactions related to TGF beta 1 is related to megakaryocytes and it's upregulated, downregulated in fibroblasts, MSCs, and some in neural cells. As a conclusion, we could highlight major players within and across phenotypes using CrossTalkeR in using two different network representations. CrossTalkeR is another package that allows a shareable and reproducible research. Here we ask you visit our page and try it out our vignettes. And as an ongoing work, we have that we are computing the p-values of these different interactions in gene cell level and also cell level. We are also doing some collaborative work with another PhD student of our group to include intracellular signaling from using a single cell toxic data, and we are using diffusion in networks to highlight the phenotype related patterns. With that, I would like to thank you all for your attention, the collaborators to make CrossTalkeR possible, all the organizers for the event for giving us the floor, and I'm here to hear your questions.